Thanks so much to all of you for being here. This is really an assemblage of, well, the top R&D heads in the industry. I want to start off, we're focusing on the consumer here today, but I wanted to start off talking about a slightly different topic. Roger has really just taken this new job over there and seems to be striking a theme that we've heard a lot from Michael and some others who aren't here, like Elias or Honey over at Sanofi. But the idea that R&D organizations can get better even as they potentially get smaller. And I was hoping you could talk to us a little bit, Roger, about your thinking here I know you're not going to give us too many details of what you're doing or planning, but just on a theoretical level, how does this work where you can lose people or monetary resources and wind up with more at the end?
Well, I'm not actually sure, Matt, that it does. That it was certainly never my goal to come into Merck Research Laboratories, a place with an extraordinary legacy of accomplishment, and shrink it with the idea that just shrinking it will make it more productive, because I don't think that's true. What I do think is true is that you have to scale the organization to meet the opportunities that exist and focus. One of the largest problems that we face in the research and development world is simply the breadth of activity that we undertake. And it's absolutely critical that you eschew distraction. That's something that we did, I think, very effectively when I was at Amgen, and Sean and I worked together there. And it's something I think that the industry is learning everywhere. And I'm sure Michael Dalston has had the same experience.
Well, Michael, what has been your experience? I mean, you obviously came into the company that when people think big in pharma, we think of you. And you've gone through some of these same kinds of struggles. How do you think about scale versus productivity? How do these things relate?
Yeah, so we started some 3 years ago to interrogate the type of questions. And I think what was one eye-opener is that R&D in the industry, and the word big just have underlined that, approach solving a problem often by adding more resources. And we wanted to embrace a paradigm where we had the right resources required, but rather looked upon some resource constraint as fostering hard prioritization and finding new ways of addressing problems. And we approached the entire challenge by looking more at return of investment than the amount of resources. Like Roger outlined, we focused on the areas where we saw best opportunity for patients and where we saw the biggest opportunity to impact outcome and deliver value, and where our company could play really well. So I think it's really about changing a little bit the equation, mixing the perspective of you need to have critical capability in your science areas, but you need also to have a business approach of making sure you can return enough value from that investment to be able to reinvest at the right time point as new areas emerge.
Sean, now switching gears a little bit, you've been talking about another way of changing your focus in a way that I think a lot of people think is somewhat risky. You've made a very big bet, particularly with Decode, on kinds of genetic information that maybe some others in the industry might think are not ready for prime time. How do those give you a better idea of the people you'll be treating and how you'll be treating them?
Well, I think that when you look at one of the fundamental issues we face in the industry in terms of our productivity in R&D, it relates to the question of the targets that we choose to pursue and pour enormous resources, so obviously time, money, opportunity cost go into a choice of a biologic target for drugs. And whether we're looking at the genetics of the tumor and understanding that as it is compared to the host, or if it's possible to understand the genetics, the human genetics of more complex trait-type diseases, obesity or schizophrenia, these sort of diseases, by a population genetics approach. If you can start with a target that is actually validated in humans, as opposed to validated in complex genetically manipulated animal models, is often the kind of validation we have, before going in the clinic. I think the pretest probability of success goes way up. And so one of the things that we've done is to...
And the example there, I think everyone here agrees that everyone's chasing is really things like PCSK9.
Things like
which is
PCSK9.
a cholesterol
Right.
gene and also very promising drug that each of you have touched.
But I think be careful here, because for PCSK9, and I don't think anyone would disagree, for any of those kinds of genetic studies, what we know is, based on powerful genetics, that if we could phenocopy that mutation in an individual and lower their cholesterol dramatically...
There is the aerobics instructor.
We would,
right?
we would reduce cardiovascular risk, provided we did that from conception.
Right.
Right? We have to do it right at the beginning. What the genetics doesn't tell us that you can intervene in a 65-year-old and reduce cardiovascular risk. It doesn't tell us that.
Right.
What it tells us is that if, in fact, you are born with that PCSK9 mutation, then over your lifetime you have lower risk. And so the real question, what has happened as a result of the genetics, push back on me, guys, if you disagree, but what has happened is that you've validated the biology, and the biology then provides an entree to the target, which is powerful.
Yeah.
You haven't validated the target.
No, right. I think the genetic information is not perfect because of exactly what Roger, there's a big difference between having, loss of function, for example, of a gene from conception and pharmacologically inhibiting it at the age of 60. That said, it's an enormous advance compared to the existing paradigm, where we choose targets largely because we view them as druggable, meaning, our technologies, our small molecule technology or our large molecule technology allows us to interdict that target, and then we build an evidentiary base around that target using often models in rodents that diverged from us 65 million years ago.
Right.
And when we put together, enough wheezing animals that can respond to the drug, we decide that it's a good drug for human asthma, go forward, spend a fortune, get into the clinic, turn over the card, and see that, in fact, this target is not relevant to humans with asthma. That is something that is happening on a daily basis in our industry. And so I think while it's not perfect, having the genetic insights is a big step forward, whether it's coming from analysis of genetic changes that occur in tumors, or whether it's looking at rare individuals in populations that have essentially experiments of nature in their genetic sequences that allow you to gain the insight that we get for something like PCSK9. But Roger's right, it's not a perfect information, but it's a lot better than what we're doing without it.
And you could say even overriding theme is to have now the opportunity to focus on human biology and understanding.
So all these drugs we've been developing were not focused on human biology in some way.
I think the challenge we had in the past was that we didn't have high throughput technology that allowed us to study such a diverse population like we are humans, and we were compelled to use much more streamlined systems in animals or cells. The problem was that we then took the starting point for long journeys on roads that were not the fastest or maybe even the best. And through genetics and a lot of high throughput biology that allow us in weeks to do things that took months to years, we can today get much more insight into the molecular understanding of human disease. Combining that with new technologies to design drugs.
New technologies. Are we at the point where, the one moving fastest is high-throughput DNA sequencing, right?
Yep.
Roger looks like he's maybe not sure, but
It's extraordinary.
Yeah.
yes.
So change the world.
Faster than any other technology.
Is that ready?
Yes.
Are you ready to employ that more in oncology clinical trials? And is this something that's ready for prime time in clinical development?
No, I think, obviously the high throughput DNA sequencing is tremendous. Oncology is a great area where you can, in maybe a week or so, get the genome from a tumor, and it would take years, a decade ago. But it's just one piece. I think we will see very soon not only the genome atlas, but also the protein atlas of every cell. And today we can express proteins in days. It took months. We can analyze their structure with our drugs in a few weeks. It took quarters. So you see the time is, and the pace allow us to approach complex problems faster and with a magnifier that I think promotes some of the enthusiasm that we see that the coming decade will offer much more opportunities for us. The last decade wasn't a lost decade. It was just much more incremental, but it builds a foundation where we can influence and get more outcomes.
You know, another place where the high throughput sequencing has made a huge difference is in the population genetics environment, because it's really been possible now to examine individuals at the nucleotide level. And at Decode, for example, we really are able to understand down to the single nucleotide level all 3 billion base pairs for a collection of samples of around 200,000 individuals. You know, this is extremely powerful, and it was completely impossible to do this just 3 or 4 years ago. So that's, you know, that's a big breakthrough.
It's also...
I want to move to a related point. With all this and other new technologies, are we actually getting more efficient at drug development, or are we still facing the same kind of curve where cost per NME just seems to increase exponentially as time passes?
I'm not sure we have the answer to that. It's hard to get it in, you know, sort of real time in the midst of it. What I would say is we are getting better at this. There's no doubt. So if I look across, having been doing this now for some long time, I would say that you know, routine failure modes that we used to see, particularly for small molecule development, are, you know, largely going away for the big professional organizations. Not completely, but we do a much better job with respect to absorption, preclinical toxicology, variation from one individual to the next in terms of exposure. Those kinds of failure modes for pills we're doing a lot better on. We are still flummoxed by biology. That's the problem. I mean, the fundamental issue is we don't really know how this machine that we inhabit works. We just don't know. We have very little idea about it. And so we can't tell what's wrong when it appears to be broken. And we have great difficulty then trying to fix it. So there's an enormous amount of additional fundamental research that's required to get to that point. And just as an example, to be a little provocative, if you think of application of population-based genomics to the understanding of disease, let's pick asthma as an example. Now, there are a half a dozen really big genome-wide association studies for asthma. There's a lot of work that's been done on predisposing factors for asthma. We have a whole set of genes that have been implicated in the pathogenesis of asthma. And if you look at the currently available therapies, some of which treat asthma fairly well, not one of the targets for those therapies was identified by genome-wide association screen. None of them. And the reason that's true is because when the pathology is evident, you don't necessarily get the greatest advantage from treating the predisposing factor. You might have to do something that induces a sort of alternative pathology that cancels it out. You know,
so in other words, our best targets may not be the things we show up in.
So all that is just a way of saying that let's be cognizant of the fact that we have a long way to go to develop a deep understanding of human normal physiology as well as pathophysiology to enable us to improve productivity in developing new drugs.
I'm going to open up to audience questions if there are any out there. We can keep going, but Sue wants to ask a question, so.
We always like Sue to ask questions.
We're starting with Tony.
Yeah. Bobby Robbins from Texas Medical Center. I just wanted to ask you how you see the use of disease-specific IPS lines in drug development.
Ooh, that's a good
I could say one comment there. I think in some areas they are really important, particularly for target types that have a complexity that's linked to the particular tissue and the human tissue that they're expressed in. One example, ion channels, very complex, multiple proteins involved, and they transmit signals that...
Ion channels as in things like CF and cystic fibrosis.
Yeah, that's another, you could say, transporter channel. Ion channels usually are involved in pain and neuroscience diseases, but clearly the chloride transporter belongs to that wider group. And the availability of patient-derived stem cells allow you to study and have much higher predictability. And of course, using, as Matt suggested, cystic fibrosis, you get the really right mutation from the patient. So in the areas where it matters, we are starting to incorporate that in our screening. And I think we start to see findings that differ from if we would have used a traditional cell, like we did a couple of years ago. We would only have been able to pick a drug with a very narrow understanding of its activity. Now we're able to differ between a number of drugs that all look the same in the previous era.
All right. Now we'll go to Sue.
So I had a question about clinical drug development. You talked about, each of you mentioned how difficult it is to predict human biology. So if you think of uncertainty as what you're trying to control every step along the way, trying to decrease how little you know as you proceed in product development, I'm struck by how many of the advances you've talked about, high throughput sequencing, using human biology rather than non-human biology, are preclinical or non-clinical. Do any of you see significant innovation in clinical development? Because Roger mentioned the uncertainty of using genetics when somebody's 65 years old. So it would be great if you had a bit more high throughput, efficient clinical development system that you could ask and answer these questions. Are there any insights that you can offer into how to make that piece of product development more efficient?
Yeah, Sue, it's a great point. I often say that we do 21st century biology in our laboratories and then do clinical trials that Hippocrates would have been quite comfortable with. And so that is a big challenge. As you know, we also have this issue that the way in which we improve our output in clinical trials is by narrowing the variables as much as possible, and you've played this game. And that means we get a very extremely well-defined set for clinical trials, but trying to generalize then to the population with the disease is more challenging. I think that at the moment, the things that have improved our ability to evaluate clinical data are actually mathematical. So we have much better statistical measures, and we have adaptive trial designs that assist us in making decisions earlier on. So we do that better. But at the point of care, in terms of actually where you have a strong interest and a strong role, at the point of care, we're not doing much better in terms of actually getting patient data and evaluating what's going on in the individual patient. And frankly, the cost of those things isn't coming down either. I don't know how others feel about it.
Yeah, I think we have some great examples. Unfortunately, they are right now more of the exceptions than mainstream. We contributed with one great example, Xalkori, for ALK-positive lung cancer. The drug would never have been identified as beneficial for patients if we would have taken a traditional path forward, as we understood that it was linked to a 5 to 7% subgroup of lung cancer patients and enriched them. We could see dramatic responses that really changed lives for patients. Similar has happened in melanoma with the BRAF mutations, lung cancer EGFR mutation, a small group. The key now is to make those exceptions to the more common paths for drug development. And to make that change, we need to come together in a larger group to allow us to develop drugs with not just one diagnostic per drug. We need to have an opportunity where there are a panel available, maybe 100 genes that determine lung cancer. That's not possible today, and it's so inefficient. Think through that you may have, with this current paradigm, you would end up with clinicians looking at 10,000 different single diagnostic kits. and being paralyzed. So modern technology, as Roger spoke to, would offer sequencing and maybe ability with one kit to look at thousands of genes. However, today there is not a path forward, regulatory path forward, that could be combining drug development with medical practice. Great universities such as UCSF, other stakeholders working with us could probably propose path forward. And I think what you heard about Xalkori or the BRAF drugs will become much more common and will open endless opportunity to also combine drugs.
Sean, what do you think? What are our hurdles to increasing efficiency in running clinical trials? I mean, we've had people try everything from better EMRs to large simple trials where you collect less data from more people.
Yeah, it's a big challenge right now. I agree that in the areas where we have the ability to select patient populations in whom we can perform smaller, more trials because we are looking for a bigger effect size because we've identified people based on some set of markers, whether they're genetic or other, is terrific. It's very difficult to do in complex trait diseases. It's fairly easy to do in infectious disease settings. It's fairly easy to do in oncology. It's becoming the norm in oncology, more or less. But I think that when you really get down to the nuts and bolts of the clinical development process, the thing that I think, at an operational level is likely to have the biggest impact over the next 5 or 10 years for the industry is the openness that's been expressed by the regulatory agencies around using a kind of a centralized monitoring approach to clinical trials. Right now, we spend an enormous amount of money putting people out in the field into clinical trial sites, examining how the work is being done in a sort of blanket way, where every site gets visited multiple times. It's extremely expensive. And we have begun piloting projects, and others have as well, to try and monitor as we collect the data from the sites, monitor the data, understand where the signals are, where people may not be following the protocol or there may be issues, and targeting the visits to the sites. And while this is rather operational, you're talking about really big dollars here, so it can have a significant impact. I actually think if you had to put your finger on one thing, Sue, that was going to affect cost of clinical trials over the next 5 or 10 years is probably that.
And we could just say that there are some initiatives like Transcelerate, where J&J, together with us and a few other companies, brought a consortium to try to take on some of the challenges that you said, to have more of a standard approach, and to avoid that investigators will have 5 different companies to train with.
You train the same way and sign in the same way for everybody.
Yeah, but GCP is GCP. I mean, there's still work that has to be done to make sure that sites are adhering to appropriate things.
How much do you think we could, if we could build a clinical trial enrollment into everybody's EMR or something, is there a way of making this this is our biggest cost in development aside from failure, right? Those are your 2 big things to worry about.
Right. Yeah. Failure trumps everything.
Failure trumps everything, yeah. You only have to fail a little less and you succeed a lot more, right?
Mm-hmm. Yeah, it seems that way.
Failure trumps everything, but do you think that these can be brought under control, Roger? Or...
I'm optimistic we're getting better at it. I think that just as Sean and Michael have said, that we're and Peter Heron for that matter, there's work that's being done at the level of contract research organizations as well as in our own clinical trial networks that's designed to try and improve throughput and reduce cost and reduce errors. And those things, just as Sean says, they're a pretty big lever for this. They don't get to the question which, Sue, I thought you were addressing, which has to do more with the quality and volume of data that we get, particularly for complex diseases. Cancers is sort of a different story because it's a cell-autonomous disease.
Absolutely.
And it gets a little bit to what you said, Matt, on the EMR. Do you see a world, particularly for complex chronic diseases? So I think targeting a tiny population and being efficient is awesome and should be done. But if we ever want to improve outcomes in diabetes, heart disease, I think we need to have a better approach for complex chronic diseases. Do any of you see a world where we could use observational data rather than randomized controlled trials to approve a drug?
Maybe you could say that short to midterm, when we are launching successful drugs, those that did not fail, we still often end up with agreement to do very large post-approval studies that give results many, many years down the line. I think that's a great area to introduce register-based studies that give real-time observations to monitor. And it, of course, allow some of these funds to go into innovate new drugs and move the paradigm forward. So I think if we could start there, and for US as a whole, we also need to look at how we could offer a much more integrated way to monitor. We heard Sean talking about the registries you have access through your partnership and after acquisition with Decode. Many other countries can monitor patients and understand how they do in real time in much larger integrated registers than what we can do here. Although this is the country that invented all kind of social network and mobile technologies. So this is another area where we can be much more effective and redirect important funds to help patients with new treatments.
But that was a dramatic question. What do you guys think? Is there a world?
I don't think so, Sue. I think not in the near term. I mean, I think that it's perfectly understandable and indeed laudable that there's conservatism in regulatory agencies because it's been burned so many times before. I think the only circumstance in which observational studies can be used for registration is when the treatment effect size is so large that it's really kind of unambiguous. And there are relatively few such, they have to be quite acute diseases where you have you're pushing on a really big lever. It's, it's Pen G and pneumococcal pneumonia. That's, observation studies work there. I don't think it works for most of the things that we're talking about. And I do think that for a while anyway, we're going to be dependent upon randomized trial.
Yeah, I mean, I think Michael's point about the use of those kind of technologies in the post-approval environment where we are in fact spending enormous resources doing big outcomes type trials is a place where that would first come into play. And as you mentioned the social networking technologies, it reminded me that another area I believe for efficiency in the clinical trial arena is the process we go through today in most areas to recruit subjects to clinical trials is an extremely expensive and painful process for everyone involved. And while if you're going to have a clinical trial where you need to treat people for a year with the drug to assess the impact, you can't shorten that to 6 months. You treat for a year. But the enrollment period is often as long as the treatment period or longer. And I have to believe that the social networking revolution that's occurring is going to allow us to get to patients who would benefit from being in trials and enroll them in trials much faster. And early things that we've been doing would suggest that that's true. Increasingly, people with these chronic illnesses are pretty plugged into sites that they visit frequently. Unfortunately, many people who have chronic illnesses are older and are not on the cutting edge of the use of these technologies. That will change over time. So again, on an operational level, I actually think that's going to make a big difference too. But I agree 100% with Roger that in most cases, we're not going to be having approval based on that.
We have another question up here. You first.
Yeah.
Go ahead.
This is Bill Zarkby. Could I just amplify on the point before, because there are two examples recently where registries have been used to conduct such studies. One, actually, the FDA just approved an alternative site for percutaneous valve replacement for cardiology based on a registry data. So I think that was certainly much less costly. This is in the post-approval process. And recently, just 2 months ago, in Scandinavia, a randomized trial using registries at a cost of less than 5% that it would cost that. So certainly, electronic health record and particularly well-conducted registries could help us in this, not only cost, but also in the efficiency.
All right. Next question, identify yourself.
Joseph Kim of Innovating Pharmaceuticals. It's been said that the pharma industry is moving towards the filmmaking industry, where a lot of the big houses are partnering with the smaller, more innovative film houses to launch the products, really using their marketing and distribution channels. And at Innovio, we ourselves, we completed a $400 million deal with Roche a couple of weeks ago. I would love to hear each of you. This is a fantastic panel to bring all of you into one setting. What your feeling and what your vision is in your R&D with respect to the external partnerships.
How much outsourcing, guys? How much in-licensing? Anybody want to answer that?
Well, I mean, it's been a long time. Every major pharmaceutical company, on the order of half of their products are in-licensed.
Yeah,
that's been true for a very long time. It continues to be true. There really hasn't been that much that's changed. There are cycles up and down in terms of the availability of venture capital and partnerships, but we're all looking all the time, even at organizations that electroporate DNA, as you do, in order to have a treatment effect. So we're all looking all the time at that sort of thing. I'd say we're very much like the movie business, except they get to hang out with cooler people.
No, but things have been in-licensed for a long time. I mean, Prevnar comes from in-licensed technology, right? I mean, that's an old product. It's not an old in-license.
Oh, yeah. Well, Prevnar, the original Prevnar was developed by Wyeth.
By Wyeth, but that was based on the in-licensed stuff from, I think, Letterly. On the hip vaccine.
I mean, it's obviously a number of legacy companies that carry a proud tradition forward. But we look upon this as more of a networked R&D organization. There are a number of key capabilities to be able to translate science to products. New technologies are emerging at a very fast pace. You can't own everything in-house. You define some areas where you want to be the leader, and you try to complement that space with a partnership. And we try to do that in a much more open and flexible way with a number of academia centers, with biotech companies. We collaborated recently with Merck in the diabetes field to develop.
And a fine thing it is too. Yeah.
So I think it's a theme, but for us it's more about being strategic, add capabilities together that can solve problems that look difficult. I think that's where we need to go. There is always some cost efficiency and find space where others do this for a living. I think that's more incremental. It's about those that have new technology that makes it possible to solve big challenges.
Okay, let's go to the next question.
Here. Vinosh Varia with Home Life Tech. So with the advent of sequencing information, we are having a lot of sequencing information now. Do you guys have any thoughts on the gene silencing-based therapeutics? As we know that now we have better delivery platforms and chemistries which can make them more efficient. So do you think that big pharma companies are inclined towards moving in the development of nucleic acid-based therapeutics?
I think that was about, so the question was using gene silencing?
Yes.
I think that it's an area that we tend to track in terms of the progress that's going on, but there are still, in my view, quite considerable barriers to delivering these type of agents into the cell types of interest and getting essentially an appropriate dose of the effect. So there are some specific circumstances that are being exploited right now where these agents can be delivered, for example, to the liver, and if you're trying to completely abolish, the expression of a gene, and you can do that in the liver, you can achieve some clinically relevant results. But it's still, in my view, a technology that is facing some pretty basic physiochemical barriers that aren't going to be overcome in the near term.
Just to build on what you said, I think you summarized it very well. Over the next decade, I think we will see a couple of successes, but mainly in rare genetic diseases, where you can get a more dramatic effect by manipulating one gene, and like Sean alluded to, particularly those that have the gene defect in an organ, where the technology may be more applicable.
Roger, what do you think of gene silencing these days?
Well, I guess the question is, what do you mean by gene silencing? If you're talking about manipulation at the chromatin level that alters transcription, I think we got a long way to go. I think that's a very challenging thing. If you're talking about something that's based on Watson-Crick base pairing that eliminates particular target RNAs, typically by taking advantage of DICE or some catalysis process, I think there will be, just as Sean and Michael have said, there will be some successful therapies over the next decade, largely in rare genetic or congenital diseases where we have some understanding of what it is that we have to achieve, and typically in a liver-targeted place, but maybe some other organs. Whether that can be broadened to the larger group of more complex diseases, I don't know.
We have another question out there?
Yeah, here. Joe Herring.
There's this guy's been getting his hand up for, so.
I've got I'm Joe Ionati from Cleveland Clinic. I'm an orthopedic surgeon. My question relates to the cost of drugs. So with these efficiencies in development of drugs, what are the drivers and what's the probability that the cost of new drugs will decrease to the consumer?
I think Michael wants to answer that.
I think the key focus is to look at what value medicines brings to patients and the healthcare system. When you look at large studies, new medicines has often led to a reduction in the spend, total spend for patients. We do not always look at those tools, and it's not always possible because we don't have comprehensive registers enough unless we do targeted studies. But that's really where we would like to go. Look at medicines that becomes a solution, not a problem for the healthcare system, where the price for medicine reflects the economical gain for payers, the medical gain for patients, and the options for physicians to provide life-saving new alternative treatments. Yes, some of those may seem, if you just look at one dimension of the picture, costly, but if you take the holistic part of the picture, I do hope that we will be able to communicate better and more transparent, that we provide value, and also are involved in understanding the economical spend that in many cases can be reduced. Vaccines is a great example how a rather simple procedure takes away tremendous suffering and healthcare spend in intensive care units. Hopefully that can happen in more areas. Of course, there are segments like rare disease where it may be perceived for the society that the drug price is high. However, you need to find a way as a society to come together to help those patients that are few but have tremendous difficulties. And if you look upon the equation of innovation being funded by many to support few, which is still a tiny part of the healthcare spend, I think we can have a dialogue that is going to continue to promote innovation and save lives.
So do you guys want to take on drug pricing?
Yeah, I mean, I just say that in reality, the thing that's going to drive prices down is the fact that we all pay such attention to the issue of the cost per quality adjusted life year now. So health economic calculations are part of what everyone considers as they take on programs very early on in development. And portfolios are culled based on the idea that it's just not going to be possible to bring forward a therapy that could be marketed at a reasonable price. So that is having a real effect on thinking. Now, the cost of producing drugs, as has been well documented, and Matt has put a lot of work into this in his column in Forbes, the cost per new molecular entity continues to be way, way too high. And all of the things that we have been talking about here with respect to trying to have a better understanding of biology and ultimately trying to improve the mechanism of assessing patient outcomes in clinical trials, those will have effects on reducing the cost per new molecular entity. But in addition, there's a lot of thinking now that goes in at the very discovery level to, gee, what is the value proposition that's represented here? And if it doesn't meet a standard, we shouldn't go forward even from the beginning. And ultimately, that will play out in terms of the cost of individual medicine.
And that's relatively new, relatively new thinking that Roger's talking about that a decade ago just didn't enter the equation as we started the product development cycle. So the product development cycle is very long, 10 to 15 years, so it takes time for this to play through. Another area where there may be reductions in costs beyond the R&D costs is in the manufacture of the biologic medicines, which are very expensive.
But I mean, part of the problem is you also have pushback from payers in a way that you didn't previously, and in terms of getting uptake for drugs. We haven't seen many really big mass market drug launches in a long time where you had a drug that was commonly taken for a common condition.
Sure.
I think Januvia would be one of the most recent examples, maybe
Right.
Pradaxa.
Sure.
So, I mean, that's got to be impacting your thinking.
Oh, of course, absolutely, and it's part of the same issue. It's the value proposition. I think it's very clear that there isn't flexibility in the healthcare marketplace, and payers simply are not going to support new drugs unless they represent a tangible, unambiguous, meaningful, value-justified improvement in the care of sick people. So all of us are focused on that issue. I mean, that's what we believe in and what we talk about every day, I'm sure. I haven't visited Michael every day in Pfizer, but I'm sure he does, and I know Sean does.
Yeah, and just to build on what you said, you spoke about more primary care-like products. And on one hand, obviously, if you deliver high value, as Roger said, there is going to be room for products and fair incentives for those products. But a number of our approaches is also to target products to those patients that benefit most, and that drives also a better value proposition, and that's certainly, I think, part of our portfolio review aspects, and very much embraced.
But, I mean, one thing that worries me for the industry, for you guys, is when it starts to sound like people are saying too much of the same thing. And right now, everybody is talking about those high values in oncology, but isn't there oncology could become the new primary care. You could really get pricing pressure on all these oncology products you're doing because you're working on similar products. I mean, how do you factor that in when you're choosing what products to, I mean, obviously PD-1, which is a really promising new immunotherapy, you've decided to go ahead even though there are competitors. In fact, you've made it pretty much your biggest bet, Roger. How do you think about that world where that competition is emerging?
Well, I think there will be competition in all of these areas, and I think that will have an effect on prices, and I think that is, that's the way it should be.
Capitalism.
I mean, that's, it's good. That's a good thing. By the way, we are all, as was discussed at the beginning, who are the patients whom we serve? Well, that's our families and our friends, our colleagues, and ourselves. We're all healthcare consumers here. So, we have an interest in seeing the value proposition improve. I know I do. And I live that every day.
Thank you very much, everyone.